Okay, so one of our objectives in this chapter is to be able to read or to gather information from a phylogenetic tree if it's already given to you. And this is a skill that is sometimes tested on standardized tests such as MCAT, DAT, PCAT, and those kinds of tests. So those of you going to professional school may see some of these. And this one has already been done. I'm using this one. The only thing I don't like about this tree is I really like to see all of the modern organisms lined up at present day, but we're going to overlook that in this tree. But what you can see is the root is here. And so the common ancestor of all of these animals would be here, and then it diverges into these different groups. And some of the clade names are listed here, like the cartilaginous fish, or they all have a derived trait of a cartilage skeleton, the ray-finned fish, the lobe-finned fish, and then the tetrapods. So tetrapods have 4 legs as the derived trait. So there's a couple of clade names or indications of some of the derived traits, but let's look at, I have some questions, and it's actually on the next slide. If you're looking at your PowerPoint, you can look at both of these at the same time. I'm going to have to flip back and forth a little bit. And so you've got this tree that's been provided to you, and you're going to have to practice. Right now we're practicing answering questions. And what you're looking for when you answer, for example, at least the first maybe 4 questions here is about close relationships. So close relationships means has a more recent common ancestor. So if you remember, let's say, imagine all these animals are lined up in a single line here on the right side of the screen. That would be modern day. And then as you move backwards, you're going back in time. And on this tree, the common ancestor is all the way back here, back in time. And so what you're looking for to figure out who has a closer relationship is when you backtrack along these lines, where do the lines meet? Where is the node, N-O-D-E? And that tells you how far back they shared a common ancestor. So what the common ancestor for everything on the tree is over here, but for example, human and mouse shared a common ancestor between those 2 groups right here. And human, mouse, and dog shared a common ancestor a little bit further back in time. So you're looking on this x-axis as how far back in time would you need to go to find the point where those 2 branches meet back together. All right, so that's your definition. This is not about you guessing what you think is the right answer. You're using the tree to tell you the answer. Okay, so I'll give you an example. Is a mouse more closely related to a dog or a platypus? All right, so we're going to go back to the tree. We're going to say, okay, a mouse and a dog, their common ancestor is right here. So let me get a pen. So mouse and dog is here. Here's where the common ancestor for mouse and dog would be. Mouse and platypus, so here's platypus, here's mouse. So on that, you would have to track back all the way to here. This would be the point where platypus and eventually mouse branches off. So because the common ancestor between mouse and dog, you only have to go back this far on the x-axis. Mouse to platypus, you have to go back further. So mouse is more closely related to dog. In that one, you probably could have guessed. All right, let's look at the next one, though, because sometimes it's not going to be what your brain is going to try to tell you. Okay, you've got to use the chart. you've got to use the phylogenetic tree. Is a lizard more closely related to a human or a frog? All right, now your brain is going to try to tell you something, but you're probably going to be wrong if you're not doing it this way. Okay, lizard to human and lizard to frog. Let's find lizard. Here's lizard. Lizard to human. Here's human. So where's the common ancestor between lizard and human? We have to backtrack to here. I'll put an X on this one. So human and lizard meet up, or this is the common ancestor. Now what about lizard to frog? It really doesn't matter which frog you pick, because lizard to either of these frogs, you're going to have to go back to here. So what this means is lizard is more closely related to human because the ancestor is not as far back in time as the ancestor between lizard and frog. It's further back in time. So lizard is more closely related to human than to frog. So that one's going to be a little bit of a surprise, right? I think most students, if they were just trying to guess, they would pick incorrectly on that one. You probably would have picked dog on the first one, but you probably would have thought frog on the second one, but it's not, it's human. Okay, so let's keep going. So sometimes it goes the way you think it's going to go, and sometimes it doesn't. So that's why we're practicing the skill. If you're not using the skill, you're just guessing. Okay, is a zebrafish more closely related to a human or a shark? All right, zebrafish. Where's zebrafish? Here's zebrafish. Here's zebrafish. Let me go around twice on this one so we see a difference. Zebrafish. And then here's all the sharks. It really doesn't matter which shark you pick. And human. Human we'll use again up here. Okay. So zebrafish to human, how far back do we have to go? Pretty far. The ancestor is here for zebrafish to human. And zebrafish to shark, no matter which shark you pick, you have to go back to here. So it's slightly more related to human than to shark, because you don't have to go back as far to reach the common ancestor that is shared with the human. You have to go back a little bit further to find the common ancestor with the shark. So I think that's going to surprise you, right? Zebrafish is more closely related to human. All right. Which group of fish is most closely related to the tetrapods? Which group of fish? So here are the tetrapods. This is where four legs evolved. So which group of fish? Your choices are going to be lobe-finned fish, ray-finned fish, cartilaginous fish. So hopefully you see that the lobe-finned fish is more closely related to the tetrapods. You'd have to go back a little further to find the common ancestor with the ray-finned fish, and even a little further to get the common ancestor with the cartilaginous fish. So lobe-finned fish. So you're just reading from the chart, and you've got to remember that time is one of the axes. Draw a star on the evolutionary tree above the location that represents the common ancestor of an elephant and a chicken. Elephant and chicken. All right, here's elephant. Here's elephant and chicken. So elephant and chicken, you backtrack, and you end up here again. Elephant and chicken. Here's where your common ancestor is. Oh, it said put a star, sorry. Star. Okay. I might have you on the quiz do like a click on the point where there is a common ancestor. So that could be on the quiz that way, because I've got to test your ability to do the skill. Draw an arrow on the evolutionary tree at the location that represents the common ancestor of all animals on the tree. So that would be at the root. You know, I don't know, put an arrow somewhere in this vicinity here, you'd be good. Okay? So this is a skill that you want to be able to do to predict what's most closely related based on where the most common recent ancestor is. And again, it's not necessarily completely intuitive. So you're just using the evolutionary tree to answer the question. All right. Now, here's the bad news. Just because two organisms seem similar, often they are similar, often they are related closely, but there are some times when the similarity is superficial and they're not very closely related. So what I wrote on this slide is that apparent similarity or superficial similarity does not necessarily accurately predict close evolutionary relationships. What I mean is there are cases where two things that look really similar turn out to not be that closely related. And we'll be looking at some examples of this throughout. Really, well, in the next couple slides, I'll give you a couple examples of this. All right, so the early systematists, it's a hard word to say, taxonomists, relied on an assumption that the greater the time that two species had been diverging from a common ancestor, then the more different they would be. And that's a reasonable concept, but there are exceptions to that rule. So we are going to explore these exceptions. There are times when the rates of evolution vary. So evolution doesn't necessarily plod along at a consistent rate. Number two, evolution is not always unidirectional. There are things called evolutionary reversals. For example, fish, things that are correctly called fish, and that's actually a particular term, fish. A particular group of fish that are similar to the lobe-fin fish evolved into tetrapods. And then one group of tetrapods evolved into a whale, which is weird because you go from something that doesn't have legs to something that does have legs and then back to something that doesn't have legs. And so sometimes you can, you look at a whale and you think, oh, that must be really closely related to a fish. But in the end, when you really start to dig out, the real evidence, the evidence did not end up supporting that theory. So turns out whales are more closely related to tetrapods than to fish. And of course, that's misleading when you don't know what is related to what. So I'm not saying that you would be able to predict this. I'm just saying that we have classified things as being closely related, and then later we learn more information, and that causes us to change our hypothesis to something more complicated. And then the last thing, this one we'll be talking about, convergent evolution. Convergent evolution is where organisms separately evolve very similar characteristics, really by coincidence, by the fact that they're both exposed to the same kinds of evolutionary pressures, and they both happen to get similar adaptations for surviving in those situations. And so then they kind of start looking similar, but it's really just a coincidence. Two separate adaptations or two separate branches of the evolutionary tree that acquire similar adaptations. Now, the thing is, usually the similarities are very superficial, so you just have to dig deep. Sometimes you just need to dig deeper, but we just want to understand that can happen. All right, so cladograms. Cladograms are typically put together based on what evidence we have. We always assume they are hypotheses. So cladograms and evolutionary trees are hypotheses. And like we said, we've put together cladograms and then realized that two things that we thought were closely related in the end are not that closely related, and so we have to adjust the hypothesis when we have new information. But they're always hypothetical. Now, they're not hypothetical in the sense we know that things are related, but what we are trying to figure out is how closely related things are. So when you put together a cladogram, usually you do it based on how many similar structures they have. And these two cladograms, the one that's called hypothesis 1 and the one that's called hypothesis 2, are really convey the same information, but they're slightly different, because hypothesis 2 shows ray fin fish and sharks as being quite closely related, whereas in hypothesis 1, ray fin fish and sharks are fairly distantly related. On this tree, time is on the y-axis. So the root of the tree is the common ancestor of everything on the tree, and then time moves forward, and the very top of all of these branches is present day. So these are slightly different hypotheses. So when you have a case where there could be two different hypotheses, you're always going to favor the simplest one. Now, what is the simplest one? The simplest one is the one that would invoke the least number of adaptations. So hypothesis 1 shows 1, 2, 3, 4, 5, 6 structures that are adaptations. Hypothesis 2 is 1, 2, 3, 4, 5, 6, 7 adaptations, events. And so you're always going to favor the simpler one. So hypothesis 1 would be favored over hypothesis 2, meaning we would start with hypothesis 1, and then if we later acquired information, we might switch over to hypothesis 2, but our working hypothesis is going to be the simplest one, and we stick with that one until we have specific information that indicates that this could not be true. All right, so we go with the simplest explanation unless new information eliminates it. All right, so that's what scientists do. And we're applying the law of parsimony, and it's also called Occam's razor. The simplest, if everything, all things being equal, you start with the simplest hypothesis, and then only if you eliminate that one do you move on to the more complicated hypothesis. And simpler means less evolutionary events in this context. All right, taxonomy. You're probably familiar with taxonomy. This one shows it, I guess, from bottom up. You probably know domain, kingdom, phylum, class, order, family, genus, species. You need to know these really well. So you may remember there is a mnemonic device: domain, kingdom, phylum, class, order, family, genus, and species. So there's sentences that people have made up. One of them is, did King Philip come over for good spaghetti? Or I have heard, did King Philip come over from grassy shores? But these sentences help you to remember the first letter of each of these. Subspecies, we can talk about that a little bit later. Subspecies is what we would call breed. Strain, type, I don't know. It's not biologically significant in my opinion, but its subspecies is not supercritical, but these ones here. Now they're showing you the specific names for the classification of a dog. So a dog is domain Eukarya, kingdom Animalia. The phylum is called Chordata. The class is Mammalia. The order is Carnivora. Family Canidae. Genus Canis. Species Lupus. Canis lupus. But in any case, one thing I want you to know is that these are not the only taxonomic groups. This is a good start, but there are groups between each of these. Did I just blow your mind? In fact, sometimes there's multiple groups in between. So taxonomists add groups when they need more groups. They just add more. I don't know if there's something between species and genus. That's a good question. But all these other ones you have. So for example, a smaller grouping than order but larger than family is called suborder. For example, a group lower than class but bigger than order is subclass. But there's a grouping above class but below phylum called superclass. But superclass is not as big as the subphylum. So we use these terms with the prefix super and sub. Sub means slightly below, super means slightly above. So you do have like superfamilies and suborders and order and superorders and subclass and class and superclass and subphylum and phylum and superphylum, but there's no subkingdom that I can think of. We do superphylum. Then there is, between kingdom and domain, they call it supergroup, which I really think is stupid. But it's called the supergroups. So I don't know why they don't call it super kingdoms, but anyway. So these are, not that you need to memorize these, but I'm just saying when you're studying taxonomy, if you start digging down into particular organisms, you'll start to see these additional groups. And so I just want you to be aware that those are like in between. Those are additional classification groups in between these standard ones. So don't be too surprised if you see it. All right. But I do need you to know these in order, because then you have a concept. If you see something that is a subphylum, you know it's below phylum but above class. So you can then take those new groups that you see and you can put them into the context of the framework that you already have learned. All right. Now, the way that these classifications are done is exactly the way the evolutionary tree is done. Everything on the branch of Eukarya are eukaryotes. They have certain characteristics. So what are the derived traits of eukaryotes? Well, a nucleus surrounded by membrane. So a nuclear envelope surrounding the DNA would be a characteristic of eukaryotes, a derived trait. Membrane-bound organelles. And then you have one branch of the eukaryotes are the animals. Of course, you have plants, you have fungus, and there's even some other groups that you probably have never heard of. But kingdom Animalia, there's certain characteristics that animals have, and those are the derived traits for that branch. And then animal splits into many phyla, 36, I think, 36 phyla. One of them is Chordata, and Chordata have certain traits. Derived traits. And then one of the groups of Chordata are mammals. They have certain traits. I think we talked about hair, fur, and milk. And then the carnivores are one of the groups of mammals, and they're the ones that eat meat, and so on. So each of these groupings is really a clade. It's really a group that has certain derived traits, certain adaptations that evolved in evolution. So taxonomy and the evolutionary tree go hand in hand. All right. So if you had the root of all life, then there's a split, and then there's another split. And this would be eukaryotes here, maybe. Eukaryotes. And then that splits, and you have animals. And then that splits, probably many times, and you have chordates, and then mammals, and then carnivores. And it keeps going. And each branch, eventually you get to dog. But each branch represents a more and more narrow group that corresponds to each of these classification groups. So, you know, here, somewhere here, you have eats meat. So that would be the carnivores. So each of these derived traits that define each of these groups is somewhere on the tree. And there is a particular path that leads to dog, and there's a somewhat different path that leads to human, and there's a fairly different path that leads to plants or particular bacteria. But everything comes back to the same common ancestor, the root of the entire tree comes together. So taxonomy reflects all the different evolutionary classifications, which really reflect all of the adaptations that could be marked on the tree. All right, I'm going to take a break, and we will pick up with homologous and analogous structures in the next segment.